Dr. Laura Piccio comes from Milan, Italy. Originally, she's an MD, PhD, but has been working in the United States for several years. She's a neurologist and a research scientist and has some very interesting research. This was something that was just in the news at the end of May, that Dr. Piccio was one of the investigators mentioned in this study. Without further ado, let me pass the mic to Dr. Piccio. Thank you.
Thank you for the introduction. So I'm really pleased to be here, and today I will talk about a very interesting topic for the role of diet in MS, which is a topic of great interest for patients, for their physician, and also for scientists like me who do research in MS. As you know, we can get a lot of information about diet and MS from different sources, but we have to be very careful because a lot of it doesn't have solid scientific evidence that will support. So this is the outline of my presentation. I will first discuss about the potential mechanism linking MS with obesity and diet. And then I will review some of the most popular diets that are commonly used by people with multiple sclerosis. And next I will discuss about some of our studies. We have been interested in the role of diet for several years now. And specifically, we have studied the effects of calorie restriction and intermittent fasting in the MS animal model. And based on this data, we have initiated a pilot clinical trial in patients with MS in which we have been testing the effects of intermittent fasting. And that's what I will be talking about. As many of you know, and as was mentioned before by Dr. Brown, MS has a striking geographical distribution around the world. MS, in particular, MS is the most common demyelinating disease of the central nervous system in Western countries and affects more women. And in particular, MS incidence and prevalence are related to latitude. And in particular, they increase with the distance from the equator. Both genetic and environmental factors are responsible of this geographic distribution. However, there are changes in MS epidemiology that have been reported in the past 50 years, which include an attenuation of this gradient, latitude gradient, as well as an increase in the ratio between the incidence in females and males, as well as probably very likely an overall increase in the incidence of MS. And all these support an important role of environmental factors, because we don't expect genetic to change over this period of time. So we still don't know the cause of MS, but we know that multiple sclerosis is a complex multifactorial disease involving the interplay between genetic and environmental factors. So through mechanisms that are still unknown, the interaction between genes and environment lead to a autoimmune reaction against a component of the component in the central nervous system, and so leads to the disease. Up to now, the major environmental risk factors for MS that have been identified are infections. In particular, infection has been, what has gained a lot of attention is infection with a virus, the Epstein-Barr virus. Vitamin D level, as previously said, are inversely correlated with MS risk. Smoking, as you have heard as well. And another risk factor that has been recognized is obesity, especially obesity early in life. And of course, this, given the close connection between obesity and dietary habits, diet is going to be, it's always been considered a potential player in the environment as being implicated in MS development and progression. So this is a list of studies that have been published in the past few years and that are supporting a role of obesity in childhood and adolescence in the association between obesity in childhood and adolescence with MS risk later on. The first study was published in 2009. They looked at two big cohorts of women, the nurses Health Study 1 and 2, and they looked at the BMI at the age of 18 and the subsequent risk of developing MS. For those of you that are not familiar with the BMI, BMI stands for Body Mass Index. It's calculated as the ratio between the weight and the height of an individual, and is a way to assess the amount of body mass of a person. So people are categorized as underweight when the BMI is below 18.5, normal weight when it is between 18.5 and 25, and overweight when it's between 25 and 30, and obese when it's above 30. In this study, they show that obese, when there was a BMI higher than 30, so frankly obese, there was an increase of MS risk which was more than twofold later on in life. And these data were confirmed in a second study in a different cohort, and the observation was also extended to men, not only women. And again, obesity at the age, overweight at the age of 20 was associated with increased risk of developing MS later on. Then, later on, other studies also looked at children, confirming the similar association. Obesity in childhood was associated with increased risk, mainly in girls, but the trend was also observed in boys. And numerous other publications confirmed this data, making pretty robust the scientific evidence that obesity is associated with MS risk. So the underlying mechanism that would explain with this association between obesity and MS risk are still unknown. However, we know that the fat tissue, which is also called the adipose tissue, it's not just a fat storage, but it's an endocrine organ that is capable of producing soluble factors called hormones and cytokines, which are collectively as referred as adipokines. Adipokines can modulate metabolic functions, and we also know that adipokines produced by the fat tissue can also modulate immune inflammatory responses. So in this way, adipokines create a link between metabolism and immune inflammatory responses. There are some adipokines that have an overall anti-inflammatory effect, meaning that they inhibit inflammation, and one of those is adiponectin. And there are also adipokines that have an overall pro-inflammatory effect, and one of these is leptin. I'm mentioning these two adipokines because we will talk about them later on during my talk. In obesity, what happens is that we have an increase of the fat tissue, so an increased number of fat cells. The fat cells themselves are bigger, and there is also an infiltration of the fat tissue with inflammatory cells. This leads to changes in the amount of adipokines that are present in our circulation. Not only the amount is different, but also the proportion of the different kind of adipokines. So ultimately, this will affect metabolic and immune inflammatory responses. And in this regard, what we know is that obesity is characterized by the release of adipokines that are shifting the balance to a more pro-inflammatory environment, which leads to a chronic systemic low-grade inflammatory state that may promote autoimmunity. This is the main hypothesis that is considered as a possible explanation for the link between obesity and increased risk of autoimmunity, but there are other possibilities, other possible mechanisms, including the fact that in obesity there are lower levels of circulating vitamin D, As we have heard, as we know, vitamin D is affecting immune responses, and this could also be another factor contributing to autoimmunity. Vitamin D levels are usually lower in obese individuals because vitamin D is a fat-soluble molecule that gets trapped in the fat tissue, so it's not available in the rest of the body to exert its own function. So, as I said, there are lots of information out there that you can get from internet, from TV, books, about potential role of diet in MS. However, we have to be very careful because there are not always good and robust scientific evidence that support this. Here I'm going to review some of the current knowledge for some dietary factors that are often mentioned as implicated in MS. One is vitamin D. So vitamin D is not properly a dietary factor because food usually supplies just a little amount of vitamin D. The major source for most of the individuals is through the skin exposure to sunlight. However, vitamin D is given quite often as a supplement as well as is present in fortified foods, so quite often is among the dietary factors considered in this. So vitamin D is a hormone that which primary function is in modulating bone metabolism, but there are lots of scientific evidence that vitamin D is a potent immunomodulant, can modulate immune and inflammatory responses. So there are pretty robust scientific evidence that vitamin D plays a role in MS. There are studies in the animal model of MS showing that the administration of bioactive forms of vitamin D are able to inhibit, to prevent the disease, as well as in humans. We have heard now several times that levels of vitamin D have an inverse correlation with MS risk. So low levels of vitamin D are associated with increased MS risk and are also associated with, in patients with MS, with more disability. However, these are all association studies, so they do not demonstrate a relationship of causality. We are still lacking that information, and what is going on? We have large clinical trials that are now ongoing, which will give this answer. In the meantime, what is recommended is just to patients with MS is vitamin D supplementation, and also because quite often in patients with MS there are lower serum level of vitamin D because of changes in their behavior, including the fact that there might be less outdoor activity, so less exposure to the sunlight, as well as for some patients heat intolerance, so they don't really stay in the sun very often. Then dietary fat composition is another big topic in terms of diets and MS, and specifically the consumption of saturated fat from animal protein versus polyunsaturated fat like omega-3, omega-6 fatty acid. So population-based epidemiological studies suggest an association between MS incidence and the intake of saturated fat of animal origin. And the first studies that suggested that the fats in the diet could affect MS risk and progression are the ones that were done by Dr. Swank in the 50s. In his study, he looked at the geographical distribution of MS in Norway and the relation to diet. And what he described is that MS incidence was higher in inland farming communities compared to the communities living on the coastal region. And they attributed this to the different diets. Because the inland communities, they were eating more animal products rich in saturated fat versus the people living in the coastal region that were eating more fish, which are rich in polyunsaturated fat. These findings were also confirmed by other studies in different areas. So, and as a matter of fact, the supplementation with polyunsaturated fatty acid that are supposed to be protective is probably the most common complementary medicine, complementary therapy that is used by patients with MS. However, what we can say right now is that the data from the clinical trial that have been done are still not conclusive. Some of the studies were not done from a methodological standpoint in a good way, and some of them were negative. So we don't have a clear answer to this question, that they could be beneficial. There was a review of all the clinical trials that have been done that was published in 2012, and the conclusion is that the data are still, there is not a strong evidence that supplementation would help in MS progression. There are some evidence that it might help in reducing the relapse rate, but this is still an area that needs more investigation. Then another dietary factor that came to our attention pretty recently is salt intake. The first evidence were done in the animal model, the EAE model, in which it was shown that mice that were fed a high salt diet had a more aggressive disease. And also in vitro cells treated with high concentration of salt were differentiating into a cell subset that is considered to be detrimental in MS as well as in the animal model. And this prompted to do some studies in humans, and there is one study that came out of Brazil that showed that a higher intake of salt in MS patients was associated with increased clinical activity and MRI activity in patients. However, more recent studies done in children did not confirm this association. So this is still, we need again more study that would corroborate, that we need to corroborate these findings. So I just want to give you an overview of what could be the mechanisms to which diet can affect MS. One first way is just a direct effect on the immune system, meaning that some dietary components can directly influence the behavior of immune cells, because immune cells may have receptors that could bind the component present in the diet. For instance, for vitamin D, immune cells they have receptors that can bind vitamin D, as well as for salt or for polyunsaturated fatty acid. Another way to which diet can influence the immune system is through the modulation of the gut flora. So this is has been studied a lot in the last 2 years, and it's again an area of very active investigation. We know that the gut flora is capable of modulating the immune system, and diet, of course, is a major determinant of the composition of the bacteria that we are hosting in our gut. I'm listing here one possible mechanism to which this can happen. Like we know that the gut bacteria can metabolize some certain components of the diet, and specifically some indigestible fibers. And this leads to the production of certain compounds that are named short-chain fatty acid, that we know that they are capable of stimulating the production of protective cells called regulatory cells. So this is a specific mechanism. I don't want to go into the detail, but people are now studying a lot where a component of the diet, which is mainly fiber, are capable to modify capable of the production of certain molecules by the gut bacteria that would modulate our immune system. And finally, another possible mechanism is just a direct effect on the central nervous system. Certain molecules that are in the diet that could reach the central nervous system and have an effect directly there. So then I just wanted to focus in the next 2 slides on the gut microbiota, because it's a very interesting topic that you will probably heard a lot. So the gut microbiota are the whole community of microbes that we host in our gut. And the function of the gut microbiota is a metabolic function. As I was saying, they can metabolize certain things that we cannot digest, and so it creates nutrients for us. And at the same time, a very important function is the development and modulation of the immune system. Basically, now it's not recognized that the largest probably immune organ is actually associated in the body, and it's associated to the gut. So diet, of course, as I said, is a major determinant of the composition in terms of number and types of the gut microbiome. The gut microbiome can modulate the immune system directly because certain bacteria components can modulate immune cells, as well as indirectly, as I was saying, because they produce some metabolites after the digestion of food components. The gut microbiota is playing a role in MS, and we have, for instance, the evidence in the animal model that germ-free mice do not develop EAE. Germ-free are mice that are kept in germ-free conditions, so they don't have any microbes in their gut and other mucosal tissue, and these mice they do not develop the disease. And actually, they regain susceptibility if you colonize their gut with specific bacteria. And then, very recently, the gut microbiome is also probably going to play, and that's something new. It's an area of investigation in MS, and there are some evidence that patients with MS they have a different gut microbiome compared to healthy controls. These are two papers that were published just last month. And they analyzed two samples from patients with multiple sclerosis and compared to samples from healthy controls. This is one of the figures in one of the articles in which you see the different colors are the different types of microbes, and they are comparing the structure of the stool samples from an healthy control compared to a patient with MS. You can see that there is a significant different composition, and then they also looked at patients that are under therapy in which the composition seems to resemble the one of healthy controls. And now I'm going to switch, and I'm going to review some of the most popular diets in patients that are used in patients with MS. So I'm not a nutritionist by training, but I will just go through the main characteristic of these diets and try to provide you with what are the scientific evidence that are related to that. So I just want to start off saying that there are solid scientific evidence that support the role of diet in general in several chronic diseases in humans, including obesity, diabetes, cardiovascular diseases, and also certain types of cancer. And despite the fact that many patients with MS are adhering to a specific dietary regimen, right now we have insufficient evidence that would recommend any of these dietary strategies in MS. So I'm going to review a list of diets that are often used by patients with MS, starting with the Paleolithic diet, which probably will be covered later by the next speaker. So the main concept of the Paleolithic diet is that our body does not handle well the modern diet, which is the result of agricultural development. So the Paleolithic diet basically advocates to switch to foods that were consumed by our ancestors in a hunter-gatherer lifestyle. The Terry Wald's diet is a modified version of the Paleolithic diet, which is very popular right now in the field of MS, and it's part of the Wald's protocol, and we will hear more about it. So the main recommendation of the Paleolithic diet is basically to eat natural foods and to avoid highly processed food, which have a high glycemic index, which means that they raise our blood sugars very quickly. The basic guidelines includes just the consumption of game, non-domesticated meats, and this is the food pyramid for the Paleolithic diet, and then they recommend lots of servings in terms of vegetables and fruit, and to avoid cereals. They also recommend the consumption of nuts. So the Paleolithic diet is high in fiber, and the restrictions are avoid, as I say, the consumption of all processed food, domesticated meats, and diet. Despite the fact that it's very popular in MS, there is not really a lot of evidence that it works. So there is only one study demonstrates a possible improvement of fatigue in patients with progressive MS. This was a small study, so there were only 10 patients enrolled and 8 that completed. It was only a single arm, and actually diet was just a component of a multimodal intervention. So definitely there are some limitations. So right now we need larger clinical trial that would assess the benefit of a palliative diet in multiple sclerosis. While there are some studies that shows an improvement or in patients on this diet, improvement of cardiovascular risk factors. Then we have the Mediterranean diet. The Mediterranean diet has been extensively studied in the setting of cardiovascular diseases and has been demonstrated to be beneficial. There are different kinds of Mediterranean diet, but all these are sort of sharing these basic guidelines. So high intake of whole grains, vegetable, fruits, legumes, olive oil as a source of fat, fish with low intake of saturated fats in terms of dairy and meat, and they also recommend a regular and moderate intake of wine, red wine during meals. There are no studies right now on the benefit of the Mediterranean diet in patients with MS, and as I say, there are actually extensive benefits on cardiovascular diseases. Then I'll be mentioning also the Swank diet. That is probably it's the first diet that became popular in the field of MS. It's a low-fat diet that specifically calls for a reduction in saturated fats of animal origin, and it recommends to keep the amount of saturated fat below the 20 grams per day, ideally around 10-15 grams per day. In the diet itself, the consumption of whole grain, fruits and vegetables are recommended, fish, while they recommend the use of low-fat dairy, and occasionally meat. So in terms of evidence in MS, this diet was formulated by Dr. Swank, again in the 50s, and it was basically administered to his patients at that point when there were no medication for multiple sclerosis. And he's basically performed a remarkable experiment because he followed this patient over the course of a few decades and published the data on observational data to see how their disease went over the course of a few decades. In his study, he basically recruited a patient that was around in the 50s, between 1948 and 1952. Around 150 patients were enrolled at the Montreal Neurological Institute, and they were put on this low-fat diet, in which the recommendation was the restriction in the intake of, in general, fats, but in particular saturated fat of animal origin. He reports that he followed these individuals, and he reports the result after the first complete report was published after 34 years, and basically see that he reported that those patients that adhered to the diet very strictly, which he called the good dieters, that were consuming less than 20 grams per day of saturated fat, compared to the poor dieters that were still reducing the saturated fat intake but was above the limit of 20 grams, the good dieter did better in terms of they had less neurological deterioration and there was less death rate in that group. So this is, as I say, a remarkable experiment that probably from which we can learn. However, there are lots of limitations in this experiment too, because it was there was no control group. So basically, for the criteria that now we use when we run clinical trial, there are several limitations on the conclusion that we can get from this. Then I just briefly mention about the McDougall diet, which is a high carbohydrate, low fat, low sodium vegan diet that mainly based on the consumption of cereal, potatoes, and then also calls for intakes of without any restriction of vegetables and fruit. It's basically a vegan diet that you don't have to consume dairy, eggs, meat, and fish. So there are no evidence right now, studies that have been published, even though I know that there was one study done at the Oregon and Health Science Center, which has not been published yet. And then there is also a gluten-free diet, and avoid all foods containing wheat, barley, and rye, and there are no evidence for a role in multiple sclerosis. So in conclusion, despite the fact that many patients may adhere to different dietary strategies, right now there are insufficient scientific evidence to recommend any of these diets. However, there are common themes in this diet, which are avoiding highly processed food, so foods with high glycemic index and food that is high in saturated fat, increasing consumption of fruit and vegetables, so increasing the content of fiber in the diet and reducing consumption of meat. This seems to be the common theme of all the diets. So more systematic evaluation of dietary strategies in MS is required, and in the meantime, when physicians recommend a healthy diet, I guess these are the common things that the patients they would need to take into account. And then I'm going to talk about the next part of my talk, in which I will basically review what we have done in terms of the effects of calorie restriction and intermittent fasting in the MS animal model. Calorie restriction, by the way, is also a dietary regimen that is now under consideration. There are few trials. One was conducted in our center, but there is at least another one ongoing in patients with multiple sclerosis. So we have the initial study that were done by our group were in the MS animal model, which is called the EAE. EAE stands for experimental autoimmune encephalomyelitis. It can be induced in mice, and it's induced by immunization. So we administer to the mice a component of the central nervous system that triggers an autoimmune reaction and a disease that in some way recapitulates what happens in patients. What is important is that several medications that are now FDA approved for multiple sclerosis were initially developed in the EAE model. So we have been interested in studying the effect of calorie restriction in the MS animal model, and calorie restriction is defined as a reduction of calorie intake below the usual ad libitum, which means without restriction intake and without malnutrition. There is quite a bit of literature suggesting that calorie restriction is associated to reduction of systemic inflammation, and the mechanisms are basically calorie restriction is at the other end of the spectrum compared to obesity. When we do calorie restriction, we have a modification of our fat tissue so that the balance is shifted towards a more anti-inflammatory environment, so inhibiting inflammation. So I'm going to just briefly talk about also like a finding an observation that we made in our laboratory that instigated also our interest in looking at calorie restriction. And what we saw is that in some of the experiments we came across specifically two mice that were very tiny, and the reason why they were tiny is that they had a malocclusion problem, so they couldn't eat very well, and they remained tiny. When we had to do our experiment, we were really worried, and we said, we were not sure, shall we include them in the experiment or not? Because of the weight, we were afraid that they would not tolerate the treatment. But actually, we decided to go ahead and immunize these mice, these tiny mice, together with all the others. And surprisingly, what we have observed that the tiny mice, instead of doing poorly, they actually did much better. As you can see here, this is the clinical course of the disease. On the x-axis is the days post-immunization. On the y-axis, the clinical score of the disease. The white circle are the control groups, so the normal mice, and this is the clinical course over time. And here you have the two mice. I just plotted the two mice. One mouse has a less severe disease, and one of the tiny mice that didn't get sick at all. So basically our conclusion is that these mice, they were inadvertently calorie restricted, and probably this inhibited the disease. So then we decided to study in a more systematic way the effect of calorie restriction, with the hypothesis that calorie restriction can ameliorate EA by activating multiple anti-inflammatory pathways. And here is what we did practically. We started with like 4 or 5 weeks old mice. They were divided into two groups. In one group, mice were just eating ad libitum, so they ate without any restriction. In the other group, mice were subjected to 40% calorie restriction. The dietary regimen lasted 4 or 5 weeks, and then mice were immunized. Of course, there was a difference in body weight, and then we followed them for the clinical course of the disease. Here is the course of their weight. So at day 0 is when we start the diet, and we check how much they eat 10 days before starting the diet. So based on how much they eat, that's when we reduce the 40%. And then we have the red line are the calorie restricted mice, the black line are the ad libitum fed mice. As you can see, the normal group, the ad libitum fed group, they just grow, while the calorie restricted, they sort of plateau. And at the time of immunization, there was a significant difference between the two weights. Here is the clinical course. We have done several experiments in different strains of mice to generalize our findings. And you can see in both experiments, the red line are the mice on calorie restriction, the black line are the ones that are fed without any restriction. And you can see that calorie restricted mice, they had a delayed onset and a less severe disease. And also a reduced incidence. Was 100% the incidence in the so-called control group, while in the calorie restriction in one strain was 63% the incidence, 88% in the other strain. So at the end of the experiment, we also looked at markers of inflammation, demyelination, and damage of the nerve fiber and in the spinal cord, and they were quantified and they were all reduced in the CR group compared to the control group. In order to try to understand the mechanism that could mediate this beneficial effect of calorie restriction, we measured in the serum of the animals different factors that can potentially modulate inflammation. First, we looked at corticosterone. Corticosterone is a steroid hormone, it's the equivalent of cortisol in humans, and it's a potent anti-inflammatory molecule. It's related to steroids that are used for the treatment of MS relapses. The serum was collected at different times during the experiment, at the baseline, before starting the diet, after 1 month on a diet, pre-immunization, after immunization, before the clinical onset, and then during clinical EA. And what we see that no difference at baseline, while corticosterone has dramatically increased in mice on calorie restriction. We also looked at leptin and adiponectin, and we see that leptin, which is pro-inflammatory, is reduced in mice on calorie restriction, while adiponectin, which is anti-inflammatory, is increased in mice that are on calorie restriction. So overall, the conclusion is that calorie restriction induces a shift, as it was our hypothesis, of the production of soluble molecule in our system that would inhibit inflammation. However, calorie restriction, especially 40% calorie restriction, would be very hard to do in humans. So that's why we decided to use a different paradigm, which is intermittent fasting, meaning that it's basically fasting every other day. And this would be probably more feasible in humans. First, before doing the study in patients, we tested again in the animal model, just to see if we were seeing the same effects as chronic calorie restriction. And we used basically the studies that design was similar, so we divided the mice into different groups. One group was just eating normally, the other group was on intermittent fasting, so they were given food every other day. And this lasted 4 weeks, and then mice were immunized, and then we followed the clinical course. This is the clinical course of EA, and as you can see, we still see a reduction of the clinical course in mice on intermittent fasting, while when we do intermittent fasting, we don't see a chronic reduction of fasting, but of the weight. The weight actually fluctuates based on the fact that the mice are eating or not when you check the weight. We also look for intermittent fasting at levels of corticosterone, leptin, and adiponectin, and we saw the same changes that we have seen with chronic calorie restriction, and we are also now looking at the changes in the gut microbiome that may contribute to the effects of intermittent fasting on disease clinical course. And based on this preliminary data, we decided to initiate a pilot clinical trial in which we are testing intermittent fasting in patients with multiple sclerosis. This is the way we have done the study. We enroll patients that are having relapse and that are candidates for steroid treatment. Patients that are eligible are randomized to one of two groups. In one group, they just do steroids and they eat their regular diet. In the second group, they do intermittent fasting, so they do steroids and then combine this to intermittent fasting. And this lasts 15 days. Intermittent fasting during this 15 days is fasting every other day. After 15 days, the patient can just quit or can end the study there, and this is called the acute intermittent fasting phase. They can quit at day 15 or they can stay in the study up to 6 months, and in this case all the patients are doing fasting. That is either a 2 or 3 days of fasting per week based on their body mass index. The main outcome, the goal of the study was to look at markers in the serum and see if we were seeing the same changes that we have seen in the animal study. So we collect, besides the clinical evaluation, we collect blood and serum before the study, day 15, month 3, and month 6. And we also collect stool samples to look at the gut microbiome. As I mentioned, we have an acute intermittent fasting phase, which is mandatory for everybody and lasts for 15 days. The chronic intermittent fasting phase is optional, and they can decide to stay for the 6 months, doing 2 or 3 days of fasting per week. When they do intermittent fasting, there is a supervision. It's done with the supervision of a dietitian that has a lot of experience with the calorie restriction in humans. And when we say that they do fasting, it's not that they fast completely every day, the whole day. They basically eat 1 or 2, they can drink non-sugary beverage without any restriction, and they can eat 1 or 2 salads per day with a light dressing. It can be olive oil or a commercial dressing. The salad needs to be made with non-starchy vegetables and can be either fresh vegetables or roasted vegetables. And the total calorie intake, if you know that they follow the diet, should be around 400-500 calories per day. And as I say, this is done with the supervision of the nutrition that is always in close contact with the patient. So we have some preliminary data because we have enrolled. This study is fully enrolled, and we have completed the first 15 days of the trial for all the subjects, and we have looked at leptin and adiponectin, which were markers that we have looked at also in mice. And we have seen that in those 15 days of intermittent fasting, the patient that were doing intermittent fasting at a lower level, a reduction of leptin levels compared to the one that were on the normal diet, while we didn't see changes in adiponectin. We also saw, as probably expected, which is in that way also reassuring, a change in the BMI of the subject, with a reduction of the BMI for the patients that were on intermittent fasting. In terms of the status of the study, as I say, is closed for enrollment because we have reached the goal of recruiting the 16 patients. It was a small study, mainly to be able to determine feasibility and to get some preliminary data for larger trial. So all the 16 patients, as I said, have concluded the acute intermittent fasting phase, and Of the 16 patients, 8 decided to continue in the chronic phase, and 2 are still doing it. Of the 8 patients that entered the chronic intermittent fasting phase, 2 were able to do it until month 3, and then they dropped out. I just want to spend, one slide just discussing about the compliance with the diet. Definitely, it's not easy to do intermittent fasting, even for the more, for highly motivated people. What we can conclude for our studies that for the acute intermittent fasting phase, we had a pretty good compliance. Most of the patients were able to do it, with just occasionally a violation of the protocol. And the hardest part for the participant was basically the fact that they were doing it in concomitant with the steroid treatment. And for some patients that was hard, especially because they were more hungry with steroids. Then, for the chronic intermittent fasting phase, it was in general harder to follow. So some patients, they liked it a lot, and some of them, they are continuing the diet even after the study. And for some patient that was taking an anti-diabetic medication, at the time of the beginning she stopped taking it because she didn't need it anymore, because there was a general benefit in her level of blood sugars. However, for some patients can be very difficult, especially those patients in which the disease is not well controlled. So, following this diet, which requires lots of efforts, was harder. And it was usually harder doing intermittent fasting, just combining intermittent fasting with the regular diet when there was a trip, holidays, or a vacation going on, activities also done with other people. And in general, 2 fasts per day were much better tolerated than 3 days of fasting, especially because having 2 fasts, they didn't have to do it exactly the same day. They could move them around based on their life. We observed, as I said, a mild weight loss that was in the order of 5-6% compared to the baseline, but there was not a major weight loss. And now we are measuring all the other different biomarkers that I was mentioning to you about in the blood. So in conclusion, this pilot trial demonstrated that intermittent fasting done in combination with steroid treatment during the relapse reduces leptin level, and this might contribute to an anti-inflammatory effects. Most of the analyses of this study are still ongoing, and we are now seeking funding to perform a larger trial in which we will compare intermittent fasting with other dietary regimen, and specifically a palliative diet and a Western style diet. And then I just wanted to briefly mention that at the center where I work, I mainly do research. This is something that the clinician shared with me. The recommendation to the patient, as Dr. Brown was saying in terms of diet, is just in general to adhere to a healthy diet with those common things that I mentioned. So mainly avoiding all processed food that have a high glycemic index, reducing the introduction of saturated fat with the occasional consumption of meat, and they encourage the consumption of fruit and vegetables, which are a great source of vitamins and a great source of fibers, which from a gut microbiota perspective, as I was mentioning, seems to be a very healthy approach. So for all the supplements that you probably will read about, there are studies that have been done, but in many cases these are not conclusive, and so there are quite often not proven benefits, with some exception, which is vitamin D that you have heard about, and also in terms of polyunsaturated fatty acids. The scientific evidence is not like they are there, but there might be some benefits that will need to be further evaluated. And finally, I just would like to thank you, the people in the laboratory that helped with the animal studies, then all the people that are involved in the human, in the intermittent fasting study in patients, the co-principal investigator of the study, which is Dr. Cox, the director of our center with which I've been working. She was my mentor, and we are now working together on this campus. And then Luigi Fontana, who is an expert in calorie restriction in humans, and that works at Washington University, and he was able to help us with applying all the protocols that they already used. And then, as you probably can tell, my country of origin is Italy, which is famous all over the world for food. And this is like an example of, very simple food which is consumed, or it was consumed, because, habits have changed there too. So fruit, fresh tomatoes, bread, which is simple food, but, sometimes the simplest is the healthiest. So, and then I thank you for your attention, and as Dr. Brown was saying, we take questions later. Thank you very much.